Hello, my dear students. Welcome you all to Sri Jain Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, today I am going to start with artificial kidney. Artificial kidney is also known as hemodialyzer. It is a machine that is used to filter the blood of a person whose kidneys are damaged. So, in case of renal failure, whether it is acute or chronic, okay, artificial kidneys, they come to be a very promising treatment, okay, way of treatment in filtering the blood of a person. The process is called hemodialysis. It may be defined as the separation of small molecules, that is crystalloids, from large molecules, that is colloids, in a solution by interposing a semi-permeable membrane between the solution and water. This solution is known as the dialyzing solution. So the process is called hemodialysis. It may be defined as the separation of small molecules known as crystalloids and the large molecules known as colloids in a solution by imposing a semi-permeable membrane between the solution and the water, that is dialyzing solution. It works on the principle of dialysis, that is diffusion of small solute molecules through a semi-permeable membrane. Okay, so it is basically based on the diffusion of small solute molecules through a semi-permeable membrane. That is the principle of dialysis. Okay, hemodialyzer is a cellophane tube suspended in a salt water solution. Hemodialyzer is basically made up of cellophane. Okay, it is a cellophane tube which is suspended in a salt water solution of same composition, that is isotonic to the normal blood plasma, except that no urea is present in this solution. Okay, so hemodialyzer, basically it is a cellophane tube which is suspended in the salt water solution, that is dialyzing solution, okay, which is isotonic as normal blood plasma, except that urea is not present. Blood of the patient is pumped from one of the arteries. The blood of the patient is pumped from one of the arteries into the cellophane tube after cooling it to 0 degrees Celsius. So first the patient blood is pumped from one of the arteries into the cellophane tube. Okay. After cooling it with to 0 degrees Celsius and mixing with an anticoagulant, that is heparin. Okay. So this blood is cooled to 0 degrees Celsius and it is mixed with anticoagulant heparin to prevent coagulation. To prevent coagulation or clotting. The cellophane pore, mean the pores of the cellophane tube allow urea, uric acid, creatinine, excess salt, and excessive H plus ions to diffuse from the blood into the surrounding solution. Okay. The blood thus get purified and is warmed to the body temperature, checked to ensure that it is isotonic to the patient's blood, and mixed with an antiheparin to restore its normal clotting power. Okay. So the pores of the cellophane tube allow urea, uric acid, creatinine, excessive salts, and excessive H plus ions to diffuse from blood into the surrounding solution. The blood which get purified is warmed to body temperature. Okay, it is checked to ensure that it is isotonic to the patient's blood, and it is mixed with antiheparin to restore the normal clotting power. This blood is then pumped into a vein of the patient. The plasma proteins remain in the blood. The plasma proteins, they remain in the blood, and the pores of the cellophane are too small to permit the passage of these large colloidal molecules, that is plasma proteins. The plasma proteins, they remain in the blood. Okay, because of this plasma proteins, the osmotic pressure is actually generated. Okay, the use of artificial kidney involves a good deal of discomfort and a risk for the formation of blood clots. Okay, so it is not that artificial kidney is very, easy way to use. The artificial kidney, it basically imposes high risk to the patient for the formation of blood clots. It may cause fever, generalized allergy, or generalized, anaphylaxis, cardiovascular problems, and hemorrhage. Kidney transplant is an alternative treatment for this. If the patient is not suited for what we call the hemodialyzer, then the kidney transplant is the only way out. Okay. The working of artificial kidney. Okay, so you can see the membrane with different pore size, the membrane filters with microscopic pores. Okay, toxins will be removed to the bladder. Okay, to the as waste. After ensuring the device, the patient's blood will pass to the two components. One is the filter side, second is the cellular side. Filter side is first silicon membranes will filter the blood, removing toxins. Cellular side, then a bed of transplanted kidney cells will regulate the chemical balance of the body. So this is the working when the machine is having filter side as well as the cellular side. Okay. Radial artery is used to take blood out, and radial vein is used to give blood in. Okay. Kidney transplantation or renal transplantation. Renal transplantation, okay, it is the process in which donors are matched, okay, donors are matched mean their HLA, human lymphocyte antigen, and their MHC is matched, major histocompatibility complex, to avoid any misreaction or rejection of the transplanted organ. Meaning, grafting a kidney from a compatible donor to restore kidney functions in a recipient is known as the kidney or renal transplantation. The recipient may be suffering from kidney failure, and this process is known as what? Renal transplantation. The first kidney transplant was performed between identical twins in 1954 by Dr. Charles Hungel, a Washington surgeon. India's first kidney transplant was done on December 1, 1971 at Christian Medical College, Vellore, Tamil Nadu. The recipient was a 35-year-old person, Sanenguan. Eligibility. All patients with terminal renal failure are concerned eligible for kidney transplantation, except those who are at high risk from another life-threatening diseases. Okay. Donors. A living donor can be used in a kidney transplant. It may be an identical twin, a sibling, or a close relative. If the living donors are not available, a cadaveric donor, mean from cadavers, mean dead persons, may be used. Cadaver is a dead body. Over half of the kidney transplants are from cadavers. Success rate. A kidney transplant from an identical twin called isogenic graft or isograft is always successful. A renal transplant from a sibling or a close relative or a cadaver termed allogenic graft or homograft is usually successful with the use of immunosuppressant drugs that prevents the graft rejection by body's immune response. Many renal transplant recipients are known to have retained functional grafts for over 20 years. Early renal transplantation was limited to patients under 55 years. Now, however, with the better techniques, kidney transplanting or grafting has been done in selected patients with the 70 years of life or 7 decade of life. Pre-transplant preparation. It includes hemodialysis to ensure a relatively normal metabolic state and provision of functional and infection-free lower urinary tract. So pre-transplant preparation, under this pre-transplant preparation, it is confirmed that the person should have a relatively normal metabolic rate, and the kidneys, the urinary tract should be infection free. Donor selection and kidney preservation. The kidney donor should be free from hypertension, diabetes, and malignancy. A living donor is also carefully evaluated for emotional stability. So everything is checked before the donor is selected. Normal bilateral renal function, freedom from when both of the kidneys are functioning, freedom from any other systemic diseases and histocompatibility disorders like allergies, type 4 allergies, or anaphylaxis. Cadaveric kidney is obtained from previously healthy person who sustained brain death, okay, but maintained stable cardiovascular and renal function. Following brain death, kidneys are removed as early as possible, flushed with special cooling solutions such as mannitol, and stored in ice solution. Preserved kidneys usually function well if transplanted within 48 hours. Okay. Nowadays, there are many policies of government in which if a person willing can donate their organs. Recipient donors. Okay, recipient donor and matching with the recipient and donor blood and different other factors. Recipient and donor are tested for three factors. Blood group. The recipient blood group should be compatible with the donor's blood group. HLA. It is a genetic marker located on the surface of leukocytes. A person inherits a set of three antigens from the mother and three from father. A higher number of matching antigens increases the chances that the kidney graft will last for a long time. Then antibodies. Small samples of recipients and donors' blood are mixed in a tube. If no reaction occurs, the patient will be able to accept the kidney. Okay. Transplant procedure. Transplantation is done under general anesthesia. Operation takes 3 to 4 hours. Cut is given in the lower abdomen. Okay. The donor's kidney is transplanted retroperitoneally because the position of the kidney is at retroperitoneum in the iliac fossa. Artery and vein of the new kidney are connected to the iliac artery and vein of the recipient. So the arteries of and vein of the new kidney are connected to the iliac artery and iliac vein of the recipient. Ureter of the new kidney is connected to the urinary bladder of the recipient. Often the new kidney starts producing urine as soon as blood flows through it, but sometimes it may take few weeks before it starts working. A week's stay in the hospital is necessary to recover from surgery and longer if there are complications. The new kidney takes over the work of two failed kidneys unless they are causing infection or high blood pressure. The old kidneys are left in place. Okay, mean the old kidneys are not eliminated. Immunosuppression. Immunosuppression means to depress the immune response of the recipient, okay, to graft rejection. Prophylactic immunosuppressive therapy is started just before or at the time of renal transplantation. An ideal immunosuppression suppresses the immunity against foreign tissue but maintains the immunity against infection and cancer. The drug named cyclosporin in such an immunosuppressant use of the antiserum to human lymphocytes is equally useful. Use of antiserum to human lymphocyte is equally a potent immunosuppressive agent. It destroys T cell mediated immune response for graft rejection but spares humoral antibody responses to maintain the resistance against the hospital acquired infections or generalized infections. Okay. Accessory excretory organs. Skin. Many aquatic animals such as hydra, starfish, they excrete ammonia into surrounding water by diffusion through body wall. In land animals, terrestrial animals, the skin is often not permeable to water. This is an adaptation to prevent loss of body's water. Mammalian skin retains a minor excretory role by way of its sudoriferous or sweat glands or sebaceous glands or oil glands. Okay, so human skin is having sudoriferous or sweat glands and sebaceous glands or oil glands to maintain the elimination of the waste, but at very minute level. Okay. Sweat glands, they pass out sweat. The latter consist of water containing some inorganic salts, chiefly sodium chloride, and traces of urea and lactic acid. It also contains very small amount of amino acids and glucose. This is the composition of sweat. Sebaceous glands, oil glands pass out sebum that contains some lipids such as waxes, sterols, other hydrocarbons, and fatty acids. So these are the excretory organs of the body like sweat glands, sebaceous glands, skin, lungs. Carbon dioxide and water are the waste products formed in the respiratory metabolism. Lungs remove the CO2 and some water as vapor in the expired air. Lungs have access to abundant oxygen and oxidize foreign substances, thus causing detoxification and also regulate the temperature. Liver changes the decomposed hemoglobin of the worn out RBCs, okay, into the bile pigments, namely bilirubin and biliverdin. These pigments pass into the alimentary canal with the bile for elimination in the feces or in the urine in the form of urobilinogen or strepobilinogen. The liver also excretes cholesterol, steroidal hormones, certain vitamins, and drugs. So, liver is basically helpful in biotransformation of drugs. So, it acts as a detoxifying center. Liver acts as a detoxifying center. Epithelial cells of the large intestine, especially the colon, transfer some inorganic ions such as calcium, magnesium, iron from the blood into the cavity of the colon for removal with the feces. Saliva, heavy metal ions and drugs are excreted in the saliva. Gills remove CO2 in aquatic animals and also they excrete salt in many bony fishes. Okay. So this is all about the artificial kidney and the excretory accessory excretory organs. So my dear students, thank you so much. Okay, for your support. If you are having any problem, please contact us. Till then, bye bye. Take care. Stay safe. Stay healthy.